Good morning, student. Then, in the yesterday class, I will told about law of segregation. There is the first law of Mendel's inheritance. It is also called as the law of purity of gametes. Then, he will propose two laws. First, we want to study about the first law, that is called as law of segregation or the law of purity of gamete. Then, to explain the law of segregation, he was used monohybrid cross. Then, this law, it will state that whenever a pair of genes foreign character, these are brought together within an hybrid. They will become what? Separate or the segregation. Segregation is what? They will become what? Separate or segregate during the formation of the gamete. That is called as what? Law of segregation or the law of purity of gamete. Once again, I want to tell you, whenever a pair of these foreign character, here I want to show the these are the two homozygous pea plants are there. Then these homozygous pea plant, one plant it having the similar type of gene, that is called as capital T. Another dot plant it will consist of similar type of genes, that is homozygous in condition small t. Okay, whenever a pair of genes, these are brought together within an hybrid. This one is what? An hybrid is there. Okay. Then whenever a pair of genes, these are brought together within an hybrid, at that moment they will become what? They will become what? Segregate or the separate. When the formation of the gamete, those genes, they will become what? Separate or segregate. And this plant also, when it will produce the gamete, then the genes, they will become what? Segregate. That is the first law of inheritance. That is, whenever a pair of genes, these are brought together within an hybrid, they will become what? They will become what? They will become the segregate or the separate during the formation of the what? Gamete. Okay. That is called as which one? First law or the law of segregation. Then first, Mr. John Gregor Mendel, he took two pure breeding homozygous pea plants. That is, Mr. Mendel, he was selected two pure breeding, not heterozygous. He will select what? Two homozygous pure breeding pea plant, and they will show in what? A pair of contrasting character. That is called what? One plant is tall, that is a tallness, and there is what? Darkness. He will choose only one single character. That is, I think he will use the monohybrid cross. That is height of the plant body. Within that height of the plant body, there are two types. He will mention the two types. That is, the one plant is what? Tallness in nature, and the plant is what? Darkness in nature. Okay. Then Mendel, he was selected two pure breeding pea plant. One plant is what? It will show what? Tallness. And the plant is what? It will show the darkness. Okay. Then the Mendel, he used the capital letter to denote the dominant gene. Mendel, he used the capital letter. Here I will show the capital T, as well as for darkness, small t. He used capital letter to denote the what? To denote the dominant gene, and he was used small t for denote what? Recessive gene. Hence, capital letter to denote the dominant gene and small letter to denote the dark gene. Okay. Then the tall plant of the parent, it having here I will show that tall plant. Yeah, that tall plant, it having the a pair of allele or a pair of genes are there. Then for the tallness, that tall plant will carry what? Capital T genes. Then as well as dark plant of the parental one. Here you will show the dark plant for the parental one. It having two similar type of genes for the tallness. So which one? Darkness. Those are what? Small t. Hence, Mendel, he selected two pure breeding pea plant. One plant is tall and the plant is what? Dark. Then he used the capital letter to denote the dominant gene, small letter to denote the which one? Recessive gene. Okay. Then the tall plant, it will consist of similar type of genes. That is what? Capital T. And dark plant, it will consist of similar type of genes, because he selected what? Homozygous individuals. Okay. Small t. Okay. Then the which one? Pure breeding tall plant. That is capital T, where it was crossed with pure breeding which one? Dark plant. That is The pure bedding tall plant, it was crossed with what? Pure bedding dot plant. Here I will show the pure bedding tall plant when it was crossed with pure bedding dot plant. Then at that moment, during the formation of the gamete, here you will show the tall plant having the genotype. Which one? Capital T. Dot plant, it will show the small t genotype. Okay. When this pea plant, when it will produces the gamete, then those gamete, when during formation of the gamete, these two genes, they will become what? Separate. That is the one gamete, it will carry capital T, and another gamete, it will carry what? Small t. That is the first law. During the formation of the gamete, all these genes, they will become what? Segregate or the separate. Hence, during the formation of the gamete, factor, he did not know about the genes. He used the term factor. Now we can call them as what? Genes. Okay. The factors, they will become what? Segregate or the separate. Hence, all the gamete of the tall plant, they will carry capital T genes. As well as all the gamete of the dot plant, they will carry what? Small t genes. Okay. Here, tall plant, all the gametes. Here, I will mention two gametes are there. Then one gamete carry the capital T, and the gamete carry the capital T. Then dot plant, it will produce the which one? During formation of the gamete, which one? One gamete carry the small t, and the gamete carry the small t. Okay, that is all the gametes of the tall plant, they will produce what? They will carry the capital T. All the gametes of the dot plant, they will carry the small t gene. Okay. Then the F1 generation, which one? We want to get what? Capital T, small t. Here, this gamete, you must just remember, this one is an pollen grain of one plant. This one is an, which one? Egg of dot plant. Okay. Then this gamete, it will carry what? Capital T. Then this which one? Gynoecium or the egg, it will carry what? Small t. Okay. Then when these gametes, they will fuse together, we want to get the fertilization. Okay. After the fertilization, in the fertilization, the genes, the capital T come from the dot plant and small t come from the dot plant. Here we want to get the what? F1 generation. Here we want to get what? And the seed, then that seed when it will grow, then it will seed, it will grow into what? New plant body. It is taller in size because which one? F1 generation, it will carry the capital T, small t. Then here in the F1 generation, all the plants, they will become what? Tall, but these are what? Heterozygous in nature, not the homozygous, because one gamete is capital T and the gamete is what? Small t. Okay. Then F1 generation, capital T, small t, tall plant, because why? It will become tall plants. In the F1 generation, we want to get the number of plants. Okay. Then those plants are tall. Why, sir? Because capital T, it is completely dominant over what? Small t. Here you will see the which one? This capital T. It is completely dominant over which one? Small t. Hence the capital T, it will use its expression by suppressing what? Small t. Hence the F1 generation plants, they will grow in the height. Okay. Hence they will become tall plant because capital T, it will use its expression by suppressing small t. Hence the F1 generation plants, they will become those hybrids are what? Very taller in size, but these are what? Heterozygous in condition. Okay. Then the F1 generation, those are allowed to what? Self-pollinate. Here you will see the which one? Self-pollination. Okay. Then we want to get the F2 generation. Then F1 plant, they will produce similar type of this or dissimilar type of this. Dissimilar type of this here. Yeah. Within one F1 generation. This is another F1 generation. You must. Okay. Then during the formation of the gamete, in the F1 generation, 50% of gametes, they will carry capital T genes, and other 50% of gametes, they will carry what? Small t genes. Okay. Then you will see also here. Then 50% of the gamete, they will carry capital T genes, and 50% of the gamete, they will carry the small t genes. Hence, then 50% of gamete, they will carry the capital T genes, and other 50% of gamete, they will carry the small t genes. Then we want to get the F2 generation. Here we want to use For showing the F2 generation, we want to use the checkerboard. That will be proposed by Mr. R.C. Punya. By using the checkerboard, here we want to get the what? F2 generation. Okay. Within the F2 generation, here we want to get the three combinations are there. Then here I want to mention this one is male. Within the male, that is I think the endoecium or the pollen grain. Two types of pollen grains will be produced. One pollen grain carry what? Capital T gene. Other pollen grain carry what? Other 50% pollen grains carry what? Small t gene. Then this one is what? Male gamete. Okay. Then here you will see the female gametes are there. 50% is of female gamete. They will carry capital T genes. Other 50% is of gamete. They will carry what? Small t genes. And here we want to do the crossing. This one is female. This one is male. Okay. Pollen grain as well as egg is there. Capital T is there. Then here capital T, small t, because why we want to earlier than the small t, we are using the capital T because dominant gene is there. Okay, capital T, small t. Then here we want to cross this one also, capital T as well as which one? Small t. Then small t, small t. Here in the F2 generation, here we want to get the three combination. We want to get the four plants are there, but the three combinations will be produced. Here capital T, tall plant, capital T, small t, it is also tall plant. Why? Because capital T, it will give its expression by suppressing small t. Here, this is also same as it is. Hence, these three plants, they will become what? Taller in size. Okay, only one plant, that is the small t, that is the recessive genes are there. The small t, this plant, it will become what? Dwarf in size. Hence, here we want to get the three combination. First combination is what? Capital T, and two plants are what? Capital T, small t, and one plant is what? Small t. Hence, here we want to get the three combinations, that is the capital T, small t. Hence, in the F2 generation, 75% of the plants, they will become what? Taller in size, and other 25% of plants, they will become what? Dwarfer in size. Here you will see the these are what? 75% of plants, they will become what? Taller in size, and 25% of plants, they will become what? Dwarf in size. Hence, within this checkerboard or the Punnett square, then here we want to see the phenotype. Normally, already you have learn about the phenotype. Okay, phenotypic ratio. Here, three plants are, they will become what? Taller in size. Three plants, they will become what? Tall. And one plant is what? Tall. Is it externally or internally? Externally. Hence, it is 3:1. That is called as what? Phenotypic ratio. Okay. Then, why this plant, it will become tall. Why this plant, it will become dark. Hence, we want to know the genetic makeup of this plant. Okay. That is called what? Genotypic ratio. That is, genotypic ratio is what? Capital T is what? Capital T. 20% of plants, they will become what? They have the capital T, and other, this one is capital T, small t. That is what? 50% is of seeds or the plants, they will carry the capital T, small t, but they having the genotype, and other 20% is of what? Small t. This one is what? Genotypic ratio. This is, I think, phenotypic ratio is what? 3:1, and genotypic ratio is what? 1:2:1. That is Mendel's first law, and it is also called as law of purity of gamete, because during formation of the gamete, all the genes of a plant, they will become what? Separate or segregate. Hence, this law, it is called what? Law of purity of gamete. Okay, this is about the first law, or the law of segregation, or law of purity of gamete. In the next class, we will discuss another law. Okay, thank you.